Hey Mustangs, in this video we're going to be reviewing translation. Now remember, translation is the second step of protein synthesis. Once again, in order to remember this, because both words sound the same for each step of protein synthesis, the process of making a protein, you have transcription, then you have translation. To remember which one comes first, just put those two words in alphabetical order and you got it. So transcription is the first step of protein synthesis where you actually rewrite the DNA instructions into mRNA, and then the second step is translation, and that's what we're going to review here. Alright, so translation on paper is really easy. You can't do translation though until transcription has been done. So here, if you transcribe and you do your good habits, after every 3 you write a line, and the reason why you write a line after every 3 is you are identifying codons. So this every 3 letters is considered a codon. So there's a codon, there's another codon. Every 3 letters is a codon here in the mRNA. So that's a key word to remember, codon. Each codon basically says which amino acids to connect together. So AUG codes for the amino acid methionine, MET. Now, in order to do translation on paper, you need one of these charts here. Unless you're a scientist who works with it all the time, you have the chart memorized, you would need one of these charts to figure out what each of these codons code for. And remember, one codon codes for one amino acid. So AUG codes for one amino acid, methionine. The next one, AGA, if we go A, so AG, find the box where they meet, and A, it's this one right here. So arginine. And you just keep on going. CCU stands for proline. So you go C, C, find the box where they meet. The last one's U, so it's this amino acid here. So the instructions that have been rewritten from the DNA into mRNA are saying exactly which amino acids to lay down in order to build a protein. Now, the importance of translation, super important to get this right. It's very important that transcription gets done right as well. The DNA has very specific instructions saying how to build your proteins. If a protein isn't built right, it can't function properly, and it usually doesn't do its job. It's pretty rare. There could be some sort of mutation that allows a protein to function better, but that's more rare. Usually if there's a mutation in the DNA, it's usually going to lead to a messed up protein. So translation makes sure that the proteins are built correctly. So they're the exact size and shape based on the amino acids they lay down. How do you know which amino acids? That's what the DNA instructions are saying. The DNA instructions are saying exactly which amino acids to lay down in what order. So when you rewrite the DNA into mRNA, the mRNA has the instructions saying to do that. And then finally during translation here, those instructions are read and the protein gets built. So with translation, it's going to happen on a ribosome, whereas with transcription, it happens inside the nucleus. So this is if you're talking about eukaryotes. So transcription happens in the nucleus for a eukaryote and on a ribosome for a eukaryote. If you're talking about prokaryotes, that's going to happen inside the cytoplasm, and they're both actually going to happen simultaneously. So they're going to happen at the same time. So as transcription is occurring, as DNA is getting rewritten into mRNA, it's going to attach to a ribosome and actually start building the protein as well. Okay. So if we take a look at the ribosomes, ribosomes are made out of rRNA and proteins, and it has 2 parts to it. So there is a small subunit and a large subunit. Okay, so a large subunit and a small subunit, and then here they come together, and right here is where the mRNA would attach, and these are special sites for tRNA to come in. So they're actually labeled E, P, and A. So if you are a fan of the environment, you know the EPA, Environmental Protection Agency. If you remember that from left to right, EPA, you actually are labeling the sites. This is where tRNAs exit. This is where they add amino acids to the growing polypeptide, and this is where the tRNAs actually come in. So this is the entry point. They get passed over here and then eventually leave. So as the tRNAs come along. Remember, tRNAs are the delivery trucks. Okay, so if you think of a truck, tRNAs are the delivery trucks. They're the ones that bring in the amino acids to drop them off in order to build the polypeptide and eventually the protein. So polypeptide, remember, is just a chain of amino acids. Once everything folds together and you have a functioning unit, then you have a protein. So the tRNAs come into these different sites and drop off their amino acids. All right, now looking at a tRNA, the key thing is the anticodon. Okay, so remember, they're simply, don't overthink them. mRNA, remember, is simply the rewritten DNA instructions saying how to build the protein. tRNAs are just the delivery trucks bringing the amino acids to the ribosomes. So when you take a look, there's several different ways that you'll see tRNAs drawn, and even different than this even, but the shape of the tRNA doesn't really matter. Okay, when you're trying to figure out their function. So the key parts of tRNA, though, are its anticodon at one end, and at this end, this is where the amino acid would be. So on this example here, here's the anticodon. Amino acid would be attached here. Now an anticodon, there's three letters. Three letters make up an anticodon. It should sound familiar because we said that mRNA had codons in it. Here, tRNA has anticodons. Okay, so here an amino acid would attach. Any one of the 20 amino acids could attach there. And then down here is the anticodon. And the connection between codons and anticodons is that if you have mRNA, let's say you have AUG, and this is the mRNA, the anticodon matches it. So the matching one should be UAC. And the whole point of this, the whole purpose of this anticodon, this is what makes sure the correct amino acids get dropped off. So I know for a fact AUG codes for the amino acid methionine, MET. So up here, only methionine would be attached. How do I know that? Well, here's the codon. To make sure that methionine gets dropped off, the anticodon must match. So let's say another tRNA comes in and it's like AAC. It won't match and it can't drop off whatever amino acids it's carrying. And it's not going to be carrying methionine. It's not going to be carrying MET. It would be carrying another amino acid, and you don't want to put that amino acid there if the instructions don't say that it belongs there. These other visuals of tRNAs, same thing. So here's where the amino acid would attach. Here's the anticodon that has to match a codon for the tRNA to drop off its amino acid. Here's an even more simplified. So the amino acid would be connected there. There's the anticodon. This anticodon clearly would have to match with a codon on an mRNA that's UUC. Okay, so again, tRNAs are delivery trucks bringing in the amino acids. In order to make sure they drop off the right ones, anticodons have to match codons, and that's how you make sure, your cells make sure they drop off the right amino acids. All right, so if we take a look at this image here, this is a very generic image of what's going on with translation. So you have this structure here, that is the mRNA. Oops, my bad. That is the mRNA. So this whole strand here. This, every 3 represents 3 bases, so that's a codon. You have your small subunit, your large subunit. These things, remember I said the shapes that we use really don't matter. So these things that are coming in to the E, the P, and the A site here, these are the tRNAs. So that's the tRNA right there. Oops, that should be a lowercase d. And then it's carrying amino acids. So you can see the tRNA comes, it plugs in here, moves over, adds its amino acid to the growing polypeptide chain. Remember, a polypeptide chain is just amino acids chained together, and you have a polypeptide chain. Once it folds up and it has an actual function, then it's officially the protein. And then once it's dropped off its amino acid, it exits out of the E site. All right. In order for proteins to get where they need to go, they have basically an address label. So about the first 20 amino acids of a protein are the signal peptide. So the signal peptide is just the first 20 amino acids. That basically tells where that protein is going to wind up. So as it goes through from the ribosomes to the rough ER to finally the Golgi, that basically is telling the Golgi where it's supposed to be shipped off to. And finally, I already said this earlier, transcription and translation happen at the same time in prokaryotes. In eukaryotes, remember pro, no, those are bacteria, they have no nucleus, no membrane-bound organelles. Eukaryotes do have a nucleus and do have membrane-bound organelles. So in eukaryotes, transcription happens in the nucleus, translation happens in the cytoplasm on a ribosome. But in prokaryotes, transcription and translation happen in the cytoplasm at the same time.